heredity. The second mono dihybrid cross. In the previous class, we have discussed the mono hybrid cross, and today, okay, and we will going to finish this chapter. Okay, so that is dihybrid cross. Dihybrid cross. What is dihybrid cross? The cross in which uh, two pair of contrasting character, or sorry, two characters, or one pair of contrasting traits is being studied. Okay, so wait a minute. Just give me a second, okay? Do we have done the law of segregation na? In the previous class? Okay.
Yes.
Yes, Shweta, why are you not speaking? Shweta, if you want, yes, we will repeat the chapter also, but actually, Shweta is saying, okay, Saima is saying that she's not sure. Okay, so let's do here.
Hello,
Okay.
sir. Hello, sir. Can you hear me?
Yeah, I can hear you. Yeah, I can hear you.
Can
I
you
can
hear
hear
me,
you. Yes, I can hear you.
sir?
Is I'm audible to you?
Sir, you were talking about something about 9:30. Can you repeat it?
Your voice is not coming. You speak a little bit louder. We are talking about dihybrid cross. Dihybrid cross, what is dihybrid cross? A cross between two plants, which I'm talking about. It is a cross between two plants having two pair of contrasting characters. Okay. Two pair of contrasting character means in which a two pair of contrasting character or two pair of trait is studied. A cross in which two contrasting character or two pair of traits is studied. Two pair of trait is studied. Okay, this is known as dihybrid cross. Got it? Means in which two contrasting characters means two things are simultaneously is being studied. Okay, like in Mendel's dihybrid cross, if I talk about, Mendel took red color and yellow color seeds along with their shape, round shaped and wrinkled shaped. Okay, so let me discuss here. One is round shape. Okay, let me make it round yellow. He took round yellow. No yellow. Take this yellow. Round yellow. When he took round yellow. Okay. Round yellow. And another he took wrinkled green. Wrinkled green. Okay. This is wrinkled green. Okay, wrinkled green. So when he cross-pollinate these two, and he round yellow and wrinkled green, when he cross-pollinate, right? Keep in mind always in the when he took the first plant, he took round yellow and wrinkled green, and he cross-pollinate them, and he cross. Okay. Pollination. Okay, when he did the cross pollination, then actually what happened? He get all red yellow. Okay, sorry, round yellow. He got all round yellow seeds. Means he get all round yellow seeds. All round yellow. All were round yellow. Okay, no wrinkle green were observed. This was the first F1 generation. Okay, this was F1 generation. Got it? Don't know what happens to it. So just a minute, we need to erase the sliders. Got it? So he in the F1 generation, he got all round yellow. So what was the gene for this one? The gene for this one was round. What happened to this? I just write it, but it was just displaying a little bit later. Okay. Round, capital R, capital R, capital Y, capital Y. And for this one is small r and small y, small y. Got it? Got it? Are you there? Is I'm
Yes,
audible
sir.
to you?
Yes.
Okay. Now, when we are just going to, this is round yellow, and this one is wrinkled green is smaller, smaller. Then he get this round yellow and gene for this one means all were round yellow. Now when he for this one for when he self pollinate this one, okay, self pollinate. So when he self pollinate this one, okay, when he self pollinate this round yellow, when he self pollinate this round yellow, okay. Then on self pollination he get round yellow, 9 round yellow, okay. Then 3 wrinkled yellow, okay. And he get 1 wrinkled green, one was green wrinkled green, okay. And another he got green round, 3 round green, 3 were round green. So when he self pointed this one in the second generation, okay, when he self pollinate this one in the second generation, what he find actually? So on self pollination, this is small r small and capital Y small y. So when he self pollinate these two, self pollinate these two, then he find 9 were round yellow, 3 were 3 were round green, okay, 3 were wrinkled yellow, and 1 was wrinkled green, wrinkled green. Okay. So when he self pointed, which he self pointed? The F1 generation. So F1 generation, this is F1 generation. On self pollination gives you F2 generation. Got it? Got it?
Yes,
Are you
sir.
getting? So what he did actually, he took the first round yellow, and then wrinkled green. Okay, wrinkled green. And when he self cross pollinate first, then on cross pollination he find
Yes.
that he find all were round yellow. There was no wrinkled green. Like in previous F1 generation also in monohybrid cross, we get in F1 generation, we get the same all tall plants we were getting. All tall plants were there. So similarly here in dihybrid cross also, when we did the cross pollination, we get the same result. All were same species. Okay, all were same. In phenotypic structure, they were all same. So you need to keep in mind that when all same results were obtained, when all same result is obtained, okay, then what happened? Tell me. Then when then it is obtained by cross pollination. By cross pollination. Okay, got it? Or in F1 generation. Got it? Got it? Understood?
Yes, sir.
Don't know where the RAM is going. Now, when he self-pointed this one, sorry, when he self-pointed this F1 generation, then he get the 9 were round yellow, 3 were round green, 3 were wrinkled, and 1 was wrinkled green. Can you see here one thing? That how it is possible? Okay, you see that we take the round yellow and we take wrinkled green only. We take this initially, we take the round yellow and wrinkled green, but these two were not there. These two were not there. These are the new. Round green we did not take. We take wrinkled green, but we get round green also in F2 generation. We did not take wrinkled green. We take only wrinkled green, but we take wrinkled, we obtain wrinkled yellow also. So these are the two new contrasting characters are obtained in F2 or recombination character we both think. Okay, so two new contrasting characters are obtained in F2 generation. You got it? You got it?
Yes, sir.
Okay, now moving to the next part here is that if I do there that how we obtain this one. So how can we obtain this one? First of all, let me explain you that round for round yellow, if I talking about that round shape is dominant over wrinkled. Okay. So for wrinkled, we take a small r, and for round, we take capital R. So this is for round, and this is for wrinkled. Okay, wrinkled. This is for wrinkled. This is for wrinkled. Got it? Got it? This is for round, and this is for wrinkled. Now you need to keep in mind that yellow color is dominant for over green. So we take this for yellow, and this for green. Sorry, this small y for green. Okay, like in when tall was dominant over dwarfness, the tall T, capital T, and tallness, tall. This was representing the tall, and small t was representing the dwarf. Similarly, when we need a dwarf plant, we need this small t both. And if we need a tall plant, we can have capital T, or we can have a small t. These two can gives you tall plants. Okay, got it? And this will gives you dwarf. You understood? Understood? Tell
Yes,
me.
sir.
Now you need to understood here that this when we need a round and over wrinkled. Okay, then if there is a small one capital R is there, round can give you capital R both, and if there is small capital R small r, they both give you round. Okay, a round shape. Okay, this will also gives you round because one capital R is enough. Similarly, for wrinkled, we need a small r both. This if we because wrinkled is not dominant, so for wrinkled we need both should be small r. Got it? For yellow, we need capital Y, or capital Y small y. This will gives you yellow color because yellow is dominant. And for green, we need both small y. This is for green because if there is capital Y, then it will gives you yellow. Got it? Got it? Note it down.
Yes, sir.
Note it down, please. Done? Noted?
Yes, sir.
Now moving to the next part is here that how we get this round yellow. Let me help you.
Done, sir.
Okay, like we have this. Draw 16 columns, 3 lines. Okay. Now this is round. This is stand for round. Okay, the way of writing is that first write capital R, capital Y here, and then small r, small y here, then same capital R, capital Y here, and then small r, small y here. Sorry, small r, small y here. Then write capital R, small y, and then write small r, capital Y. Opposite of this one. Same here. Capital R, small y, and then small r, capital Y, like this. Okay, got it? Got it?
Yes, sir.
Then what you will going to do here is when you multiply capital R, okay, with capital Y. Okay, so what you will get capital R, capital R, capital Y here. When these gametes get fused together, then they will give you capital R, capital Y, capital R, capital Y. So you will get capital R, capital Y, both capital R, capital R, capital Y, capital Y. Here what you will get? Tell me. Capital R, then capital Y, small y. Okay, got it? Then capital
Yes,
R,
sir.
small r, capital Y, capital Y. Then capital R, small r, capital Y, small y. Got it? Got it?
Yes, sir.
Here, what you will get? Capital R, capital R, capital Y, small y. Then capital R, small y. Then capital R, small r, capital Y, small y. Then capital R, small y, small y. Got it? Got it?
Yes, sir.
Then capital R, small r, capital Y, capital Y. Then capital R, small r, capital Y, small y. Then small r, capital Y, capital Y. Then small r, capital Y, small y. Here we get capital R, small r, capital Y, small y. Capital R, small r, no, capital R, small r, yes, small y. Then small r, capital Y, small y. Then small r, small y, small y. Got it? We'll get this. Now you know that what is the round uh, yellow. If I talk about round yellow, for round yellow, round is dominant over wrinkled shape. So small one capital R is enough for the round, and yellow is dominant over green. So one y capital Y is enough to bring yellow color. Got it? Got it? So one capital R and one small y, at least one. So both are capital, both are y. So this will gives you round yellow. This is one. Okay, got it? Here one capital Y. This is also give you round yellow. This will also gives you round yellow. This will also gives you round yellow. Then this will also gives you round yellow. This will also gives you round yellow. This will also gives you round yellow. This will also gives you round yellow, and this will also gives you round yellow. Got it? So one capital R, at least one capital R and small y will gives you round yellow. So how many are you getting here? How many are you getting here? You will getting here 3 and 9. So how many are you getting here? 9. 9 are round yellow. Got it? Got it?
Yes, sir.
Then wrinkled yellow. If I talk about a wrinkled yellow, for wrinkled yellow, at least you need for wrinkled yellow. Okay, wrinkled shape is dominant. You can get the wrinkled shape if there is both small r and at least one capital Y is there. because wrinkled is at least if there is one capital R, then you will get round. So if you get, in order to get the wrinkled shape, you need both R, small r, and in order to get yellow, at least one capital Y should be there. Got it? Got it? Then how many are there for wrinkled yellow? Tell me. So
3.
here 3. Then both small r and one capital I. This one. Okay, we are just representing this one red color. Okay. So this one is both small r and one capital Y. At least one, both small r, one capital Y. So this is 3. Okay. Then we have wrinkled green. It's not wrinkled green. We'll take round green first. Round green. And it is represented by, let's suppose we are just representing it with blue color. Okay, so both for round, at least we need one capital R, and for green, we need both small y. Got it? Because round is dominant, so one capital R is enough, and green is not dominant over yellow, so we need both small y. So both capital R, one capital R, and both small y. Here, both both small y and one capital R. Here, both small y and one capital R. So this is 3, and the remaining one is round green. Okay, sorry, wrinkled green. Wrinkled green. So for wrinkled green, we need only one here, because for wrinkled is also not dominant and green is not dominant. So for wrinkled, we need both small r, and for green, we need both small y. So this is this one. Got it? Understood?
Yes.
Understood? Have you noted this one? Have you drawn this structure? Have you drawn this table?
Yes.
Everyone, you understood?
Yes, sir.
Okay. So let's do this one. We are going to have, now we have round yellow. How many round yellow? 9. We have wrinkled green. How many wrinkled green we have? 1. Okay. Then we have, these were the original that we're taking. Okay. But we are able to get the two new recombinant character, two new recombinant and characters or traits, two new recombinant traits are here. One is round with green that we have not taken, and wrinkled with yellow. Okay, so this goes with this one. If I talk about this round green, okay, so this is round and this is green. They both join together, and similarly wrinkled, and similarly wrinkled yellow. This is wrinkled yellow. So this wrinkled, this is moving with this one. Okay, so what means these two join together, they do both mix together. So how it is possible? What does this dihybrid cross conclusions we are going to have? That for every trait there is a separate gene. What is the conclusions of dihybrid cross? That there is a separate gene for round and yellow both. Okay, for example, if I'm talking about that, there is a special gene that control both round as well as yellow. Round as well as yellow. This is single gene. Okay, like this is also round. This is a special gene that I'm just showing you, that it is controlling both round and yellow. And this is the gene that is controlling both wrinkled green. Okay, wrinkled green. Wrinkled green. Both same. Means this gene is controlling wrinkled as well as green color. This gene is controlling both round and yellow. So when they cross together, they will gives you what? Round yellow. Okay. Similarly, this when cross together, this will gives you round yellow. This when cross together, it will give you round yellow. And this when crossed together, this will gives you round yellow. Because this gene, round yellow gene is dominant over wrinkled green. So we are just getting the same result that we get in the first generation. Like here, we were getting the same results. What we are getting here, that in F1 generation, here we are getting all round yellow. We are just assuming that there is a single gene that control both round as well as yellow color. Round as well as yellow color. Both it is controlling. Got it? Got it? Got it? There is a single. Now, if we are just crossbreed this one, like this one, when crossbreed with this round yellow, when it is self-pollinated with this one, this is also round yellow, this is round yellow. This gene controlled round yellow, both. This gene controlled wrinkled green, both. So when this is going to meet together, it will gives you round yellow. This will gives you again round yellow. This will gives you again round yellow, and this will gives you wrinkled green. This will gives you wrinkled green. What you will observe here, what you are going to observe here, you will observe here that this 3 will gives you round yellow. We are just assuming here, just keep in mind that we are assuming that this is a special gene that control both the round as well as yellow. So you will observe that if it is possible that only a single gene is controlling both round as well as yellow color. Okay, both round as well as yellow. Got it? Then in that case, you need to keep in mind that if both the genes are controlling, means this gene is controlling both round yellow. So in the first generation, you will get all round yellow. But in the second generation, you will get out of 4, you will get 3 round yellow and 1 wrinkled green. You will not get any new contrasting character. You will not get any new contrasting character like here you were getting. Like here you are getting the 2 new round green and wrinkled. So it is only possible if we have a separate gene for round yellow and wrinkled green. Like for example, like I'm saying that this is a specialized gene, okay, that controlled round and yellow color. Okay, this is a special gene that control okay, let me make you. This is a special gene that control round and yellow color. Okay, round and yellow color. Okay, and this is a special gene. This is a special gene that control wrinkled and green color. Wrinkled and green color. Wrinkled and green color. Okay, if this gene is same, that is controlling round as well as yellow and wrinkled and green, so they are means if the single gene is controlling both, round yellow is controlled by this and wrinkled green is controlled by this, then we are not going to get any new contrasting character. But if there is a separate gene, that this is for round yellow and this gene is for, let's suppose this gene is for, this is for round, okay, and this is for green, okay, and this is for okay, so and this is for, let's suppose this is for, okay, so if I took about this one, that this gene is controlling, this gene is controlling the yellow color, and this gene is controlling the round shape. This gene is controlling the green color, and this gene is controlling the wrinkled shape. So if they are the separate gene, then you can rearrange themselves. Okay, first order can be like this. They are having. First order will be like this. They are having. Okay, the second order, they can have a separate order. They can have a separate order. How? How can they have a separate order? Then let me show you how they can have the separate order. Okay, if they are the separate, so you can put it here. Okay, and you can place this one here over it. Okay, so this gene can be placed over the another gene. Got it? And similarly, this gene can be placed over this one. So if they are the separate gene, then they can be placed over one another, and a new contrasting character is possible. Got it? So it is only possible if we have the two separate gene. Understood? Understood? Understood, Saima? So it is
Yes,
only
sir.
possible if we have the separate gene for each character, for each trait, sorry. For yellow, for yellow, we have separate gene. For green, we have separate gene. For green color, we have separate gene. And for yellow color, we have separate gene. For round, we have separate. Wrinkled for we have separate. So how this gives the law of independent assortment. Okay. Shweta, you got it? You get this one? Shweta, you got this one? What is law of independent assortment? So what does law of independent assortment say? Law of independent assortment. So what does law of independent assortment says? So it says for every trait, there is a separate gene. For every trait, there is a separate gene. Okay, or we called it factor. Okay, and it is free to assort itself independently. It is free to assort themselves independently. Got it? So it can assort themselves. Means for every trait there is a separate gene, and they can rearrange themselves. This can come here. This can come here. Got it? Got it, Sama? You understood?
Yes, sir.
So noted this one. Noted?
Sir, I'm writing.
Okay, write it down. Okay, you can join it, Shweta. You can join it again at 9:30. Okay, if you want, if you are not able to get the proper, means if there is a communication skill lack, or we can say that if there is internet issue, then you can join it again at 9:30. Okay,
Done, sir.
should I move forward?
Yes, sir.
So the question comes in examination that how do Mhm. how do Mendel's experiment Mendel experiment shows that shows that okay, so how do Mendel experiment show that traits are inherited independently. Independently. Okay, how do Mendel experiments show that traits are inherited independently? So what's the answer for this question? that in Mendel dihybrid cross, we did the experiment with round yellow and wrinkled green, but we get the new contrasting characters or new recombinant characters that were not taken. Okay, this is only possible if we have a separate gene for each trait. Okay, got it? There is a separate gene for red and separate gene for wrinkled and separate gene for green and separate gene for yellow, and they are able to rearrange themselves in independently. Okay, so note it down. In Mendel dihybrid cross, in Mendel's dihybrid cross, the two new contrasting characters were seen. Okay, which was not taken, i.e. round green, i.e. round green and wrinkled yellow. Wrinkled yellow. Okay, this is only possible when there is a separate gene for each traits and they are free to assort themselves independently. Note this one first.
Done, sir.
Done. Okay. You also, Shweta? Done?
Yes.
Okay. Now there is a blood group concept also, that how the blood groups are being studied. Okay. So blood group, there are the four types of blood group. One is A blood group, A, then B, then AB, and then O. A blood group is when we get IA, or IO. B blood group can be when we have IB, or IO. AB blood group is when IB is there, and blood group O is when IO is there. Note it down for blood group. Blood group. Done?
That's all.
So if a person is going to have A blood group, so okay, so this is the possibility that this is homozygous A, this is heterozygous A. This is homozygous B, this is heterozygous B. This is AB is always heterozygous and this is homozygous. So a question comes in in board examination or sometimes. This is a man with blood group A. With blood group A marries a woman. A woman with blood group okay, O. O. And their daughters has blood group O. Okay, this information okay, is this information enough to tell you which of the trait blood group A or O is dominant. So what you will answer? A man marries a blood group A, marries with a blood group A. Okay. A man with blood group A marries a woman with blood group O. Okay. And their daughter is going to have blood group O. Is this information enough to tell you which trait is dominant or recessive? A or O? So no, it is not. The condition for A is actually, if we have condition for A, if a father is going to have homozygous A, condition for A blood group, or if a father is going to have heterozygous A, and mother is going to have for B, IB condition for B, it is I sorry, for condition for O, this is IO. Okay. Now if father is going to have homozygous A and mother is going to have homozygous A, then this will gives you IO. 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 There is no chance of getting blood group O. But if the father is going to have heterozygous, tell me. If the father is going to have heterozygous A, like IO, and mother is going to have O blood group, then it is going to have IO, and IO, and then IO. There is a 50% chance when the father is going to have, okay, when father is going to have condition 1. In condition 1, there is 100% chance that the offspring is going to have blood group A. But in condition 2, there is 50% chance that she will get a blood group A or she will get a blood group O. Okay, here is 50. In this one, there is and condition 1. Condition 1, when father has homozygous A. Homozygous A. Then there is 0% chance that daughter will have blood group blood group O. Got it? Sorry, blood group A. Sorry, yeah, no, 0% chance for blood group O. She will not have blood group O. But in condition 2, are you writing this one? But in condition second, we can say here in condition second, when father has heterozygous A, heterozygous A, then there is 50% chance that daughter will have blood group. Thus, since in first this data is insufficient, which is dominant or recessive blood group. Got it? Because in this second case, if we assume, then there is 50% chance. Got it? Got it?
Yes, sir.
Have you noted this one?
Sir, I'm writing it.
Have you noted the previous slide? Both?
Yes.
Note it down fast. Done?
Sir, one minute.
That's it.
Done, sir.
Now the next is sex determination. Sex determinations, actually, if I talk about that, there are two factors that are responsible for the male and female, okay, in the animals. Okay, there is some temperature in some, there are some environmental factor also that affects the, that can tell you, means that can result in the male and female, okay, like incubation temperature, that high incubation temperature uh, in some animals results in male, while in some other animals, high incubation temperature means the temperature that uh, uh, an animal provides to the eggs uh, before the kids or the babies coming out or hatching out from the egg. So incubation temperature, we call it incubation temperature. Okay, so there the sex discrimination is done by two factors. One is the environmental factor. Okay. Factor. Okay, the next one is genetic. Okay, like I'm talk about that in some animals, okay, the temperature, the incubation temperature, we call it incubation temperature. Incubation temperature means the temperature that is given to the eggs before the baby comes out. Okay, so this is high incubation temperature at which the fertilized eggs are kept decide the gender, decides the gender. Okay, but in human males, if I talk about in some animals like human gender, okay, or individual in some animals like human gender or individual is determined by a pair of chromosome, by a pair of chromosomes called sex chromosome. Okay, Zainab, are you there? Zainab, are you there? You understood? Noted this one? Noted this one?
Yes,
Now,
sir.
in human male, we have 44 autosomes, and one X and one Y chromosome. This is called sex chromosome. And human female, we have 44 autosome, and one pair of X and Y. This is called sex chromosome, both XX. That's why this is the 44 into 46. Then wait for 5 to 6 minutes. We're going to end the class because this is the last topic that we need to teach them and the chapter is over. Okay, so we will give you extra time. Just wait for 1 or 2 minutes. Okay, just going to finish this one. So here we have 44. Okay, now when the gametes are going to form, the gamete is going to have half number of chromosomes. So in human males, the sperm is going to have 40 out of 44. Half of 44 is 22 autosome. 22 autosome and either one X. This is 23. And another sperm is going to have 22 autosome and one Y. Okay, so that's why human sperms are considered to be as heterozygous. Heterozygous means they are not going to have the same. But human female chromosomes are considered to be as homozygous. Why? Because they all have the same. Means all egg is going to have 22 autosomes and one X chromosome. Okay, sperm is going to have one X, one type of sperm have X, another type of sperm is going to have Y chromosome. So when this is going to fuse with this one, then the zygote is going to form, which is going to have 44 autosomes and one X and one X other. And if this is going to fuse with this sperm is going to fuse with the egg because egg is of same types. So we are not going to make another egg here. So this will again give you 44 autosome and one X and one Y. So when there is an X and Y, this is a male child, and this one is a female child. female child. And you need to keep in mind that human sperms are responsible for determining the characters. Okay. So this will be going to write in this next class. Okay, this is going to end this one. We have just almost end this one. Okay. So, and you have you got the notes that we have provided you of bio, the PDF notes?
No, sir.
Okay, we'll provide you. Okay. So this is the human males are responsible. So there's so many questions comes in the examination that can you determine that why human male, can you explain that why human males are responsible for determining the sex of a child? So because human males are going to have X and Y chromosome. So if the sperm fuses with this one, with sperm with X chromosome fuses with this egg, then it will be a girl child. And if the Y sperm fuses with this one, then it will be a male child. So human males are responsible, not the female are responsible for the, because they are not going to have Y chromosome. They have the same. So they are homozygous. Got it? Okay. So that's all for today. You can leave. Okay, we'll give you the next one. What are you saying, Sweta? Sir, I'm not able to access after the class test. I don't know what you are saying.
So